My name is Natasha Dudek. I'm a graduate student in the Shapiro Lab at UC Santa Cruz. The big picture of what I'm studying is more about really weird lineages of bacteria that we don't know anything about. And so most people, when they think about bacteria, they think about bacteria that make them sick. But there's actually a lot of bacteria that are really important for keeping you healthy or for keeping nitrogen cycling, carbon cycling, etc. Going on. And a huge percentage of bacteria have barely been studied. So for example, traditionally the way we study bacteria is by culturing them in a lab, but only about 1% of bacteria in the world can be cultured. So there's this 99% that we don't know what they do. We pretty much know very little about them other than that they exist. So I'm from Montreal in Canada. My parents are both in science. My dad's actually a professor. And I guess the way I got really interested in biology was from watching documentaries about marine biology when I was a kid. And I watched them and I'd see like all these animals underwater and I'd just be like, oh my God, like that's what I want to study. So when I was 8, I was like, I'm going to be a marine biologist because I decided that I wanted to live on a beach and study whales and sharks and all the rest of it. And from there, I sort of decided to go into science and I got really interested in particular in studying coral reefs. And a lot of coral diseases are actually caused by bacteria, unsurprisingly. And from there on, I got really interested in microbiology, and now I'm here. A lot of what I do is on the computer. I do a lot of more bioinformatics-like research. But so in general, for my samples, other people collect the samples for me and they ship them over. The samples that I have actually come from the mouths of marine mammals. And in particular, right now, I'm working with samples that are taken from the mouths of dolphins down at the Navy because they have a marine mammal program. I'll do a bit of lab work and extract the DNA and prepare those samples to ship for sequencing. And then I get the sequencing results back through the computer, and everything else is using various computer programs to try to assemble genomes and learn more about what proteins and functions are encoded by the genomes. For example, in the human body, there's about a ratio of 10 bacterial cells for every single human cell, right? Which means that there's a ton of bacterial genes in the human body. And a lot of those genes and the bacteria that live in the body, we have no idea what they do, and we just know nothing about them. And same goes for other organisms and other environments. There's a lot of bacteria out there, and we don't know what they do, we don't know what genes they have, etc. And so learning more about these bacterial lineages that are very poorly studied can help us learn about how the world works and, in general, what bacteria do in their environment. A lot of what I do is on the computer, and it's very much like a puzzle. So a lot of times, for example, you might just have a lot of problems like getting one particular program to work. And it's like, I need to figure it out. Or I might be like, oh, that's so weird. Like, I don't know why that happened like that. And I just like want to figure it out. Yeah, it's very exciting and I like it. I think that it's really important to try to get experience early on and, for example, volunteer in a lab. That's how I got started here, is volunteered in one lab and then another, and I kind of figured out what I liked, what I didn't. I think it's also really important that you always really do the things that you like and not, the things you feel you have to do maybe, right? Like in the end, it's going to be your life and you have to kind of, go with a lab that you're really interested in and figure out what you really want to do.